Hello everybody, Arashikitsune here, and today we are playing Plague Inc. Involved, a game by Endemic Creations. There will be a link in the description below if you want to check it out. Now, I do have some hours in this game. In fact, I've used it in tutoring groups before to demonstrate concepts from epidemiology and viral evolution. But what I wanted to do in this video and maybe some other videos down the line, I don't know, we'll see how it goes, is give a brief background on the organisms in the game and the human immune response. Sounds kind of interesting, right? Maybe? Because infectious diseases are so entertaining. Anyway, let's get this show on the road. So, as you can see here, this game does allow you to experiment with a few different organisms, bacteria, viruses, fungi, Neurax worms, parasites, prions, as well as the Necroa or zombie virus, nanoviruses, bioweapons, all sorts of different stuff. But the first one that you have access to is bacteria. Now, bacteria are prokaryotic microorganisms, typically a few microns in length, and are some of the first organisms that appeared on Earth. There are an estimated 5 quintillion bacteria on the planet in all different environments, and what was I just talking about? Oh, and in humans there are an estimated 30 trillion bacterial cells in our general microbiota. No. With most of these local flora residing in the gut or on the skin. Now, the majority of these are harmless. Clear you. Due to the immune system. However, there are several species that are pathogenic, including, but certainly not limited to, things like cholera, anthrax, and bubonic plague. As mentioned earlier, bacteria are prokaryotes, meaning they lack a nucleus and rarely contain membrane-bound organelles. Which one of these do I want to go for? Let's go aquasite. Zero file. There we go. Now, the perks that you've seen me picking here, you gain them throughout the you unlock them throughout the game as you complete. Why did I do that? As you complete different modes, you unlock different things that sort of change the gameplay up a little bit. We're going to go ahead and go with normal. And what do we want to call this? And we'll just go with the general Hex 20. So, welcome to Plague Inc., your new bacteria. To win, you must evolve and spread across the world, wiping out all humans in the ultimate plague. Select a start location. I When I'm choosing start locations in this game, I tend to go for Egypt, just because it's a central hub. There's a lot of access. You've got airports, you've got harbors, and it's very centralized, so it will actually spread decently quickly. Let's go ahead and start there. And those little orbs that pop up are points, which you can use down here to evolve your disease. So I'm going to go ahead and evolve this so it will travel faster by plain and in arid climates. So going back to what I was talking about earlier, the human immune response. In regard to interactions with humans, bacteria are typically mutualists or pathogenic. For example, the thousands of species in the human gut contribute to immunity, synthesis of vitamins, the conversion of sugars to lactic acid, and the fermentation of indigestible carbohydrates. The presence of this gut flora also inhibits the growth of potentially pathogenic bacteria through competitive exclusion. Now, pathogenic bacteria interact with the host in a parasitic format, and each bacterium has characteristic interactions with the host. For example, Staphylococcus and Streptococcus bacteria, which are bacteria that exist on your skin and nasal passages every day, can cause skin infections, sepsis, pneumonia, or meningitis. Others are obligate intracellular parasites, meaning they cannot reproduce outside of a host cell. These include things like the bacteria which produce typhus and Rocky Mountain spotted fever, depending on the species, as well as chlamydia, which contains species linked to pneumonia, urinary tract infections, and possibly coronary heart disease. Now, some species are also opportunistic pathogens, primarily causing disease in those affected by immunosuppression. Now, the body does have some innate defense against pathogens, including the skin, mucosa, macrophage activation, NK and T cell activation, neutrophil activation. Oh, can't talk today. And dendritic cell activation. As I mentioned somewhere earlier, I think there are two kinds of bacterial pathogens, extracellular and intracellular. Extracellular bacteria tends to accumulate in the interstitial regions of connective tissue, the lumens of the respiratory go ahead and bump up resilience. Next, probably going to go for drug resistance, because soon they might, yeah, because they're starting to treat with antibiotics in some areas. What was I just talking about? It's the problem with trying to do a lot of this from memory. you lose track of where you are in what you wanted to talk about. Oh yeah, the extracellular bacteria. They tend to produce proteins which penetrate or cleave components of the mucosal epithelium, allowing access to the underlying tissues. Let's go ahead and bump up drug resistance. And a variety of extracellular bacteria enter the microfold or M cells in the follicle-associated epithelium. The symptoms produced by extracellular bacteria are attributed to the toxins that they produce. Exotoxins are toxic proteins that are actively secreted by gram-positive and gram-negative bacteria, and endotoxins, which are lipid portions of the lipopolysaccharide molecules embedded in the walls of gram-negative bacteria and are go ahead and bump up genetic hardening. Evolve you, make you more susceptible to survive in all climates. Anyway, the endotoxins are not secreted but released when the cell walls are damaged. So as the body starts to fight the infection, it actually releases more and more of the toxins, causing more damage. Some gram-negative species produce both endo and exotoxins, and as interesting as it is, I'm not going to go into the mechanisms of how the different toxins create symptoms. I don't really know if anybody else would be interested in that. I know that I am, but that's just me. Let's go ahead and bump up drug resistance again, because they're going to find it at some point and start treating it. So because extracellular bacteria cannot hide within the host cells, antibodies are fairly effective against these species. Polysaccharides present within the bacterial cell act as T1 antigens for the activation of B cells, while other bacterial components supplying TD antigens induce a Th2 response that provides aid to the B cells. Immunoglobulin cannot talk today. Antibodies flood the immune system, and IgG antibodies protect the tissues by binding to the bacteria, preventing attachment to the host cells. These antibodies can also act as opsonins, which coat the bacteria so that it is engulfed in phagocytic leukocytes expressing FCR, where they are vulnerable to destruction by pH changes, defensins, and the ROIs and RNIs associated with respiratory bursts. What was I just doing? Sorry, getting a little distracted. Antibodies that are made against exotoxins are called antitoxins. These neutralize a toxin by preventing it from binding to the cell it would otherwise damage. There are also the three pathways of complement activation. The classical cascade, where antibodies of a particular isotype, particularly IgM, bind to the C1q. Then there's the alternate pathway, which is activated by peptoglycan in gram-positive cell walls, or LPS in gram-negative cell walls. Last is the lectin pathway, which is activated by the binding of MBL to sugars on the cell surface. These pathways lead to the destruction of bacteria through MAC-mediated lysis. Now, as with any system, there are faults. Extracellular bacteria have evolved strategies to evade the immune system. In the case of antibodies, some bacterium ensure adhesion to the host by changing the amino acid sequence. Now go to transmission, bump up water, bump up to an extreme bioaerosol, because I want to ensure that Greenland here is infected. This is one of the hardest places to infect, surprisingly, because it only has one port of access. So you really want to make sure to catch this before the disease is discovered and they start closing down borders. Anyway, in the case of antibodies, some bacteria ensure adhesion to the host by changing the amino acid sequence of the proteins used to stick to the host surface. Other bacteria produce proteases, which cleave the antibody proteins, rendering them useless. For avoiding phagocytosis, the polysaccharide coating, which encapsulates the bacterium, produces a charge that inhibits the binding of phagocyte receptors. Yay, Greenland is infected. And in regard to the complement pathways, some extracellular bacteria can avoid these by virtue of their structure, with external protrusions that prevent the binding of C3b or the assembly of the MAC structure. Some bacterium also produces compounds which block the complement cascade in various stages, and there are others that produce blocking antibodies which compete with the complement fixing antibodies, reducing the MAC formation. Now, in regards to the game, I'm not bumping up. As you can see in this tab, you can start to increase symptoms. I'm trying to avoid doing that, because the longer your bacteria goes undetected, the more people it infects, and the easier it will be to do a systematic wipeout, which is why I'm also going to devolve the insomnia symptom that just evolved. Yes, go ahead and devolve that. Because we want to remain undetected for as long as possible, infect as much of the population as possible, and then start hitting hard with symptoms. So the other type of bacterial pathogen are the intracellular bacteria, which access the host through breaches in the mucosa and skin, though some are introduced to the bloodstream by the bites of vectors such as mosquitoes or ticks. Once inside the host, the bacterium evades the previously mentioned defenses by moving directly into the host cells to reproduce. Popular targets include, but are not limited to, epithelial cells, hepatocytes, and macrophages. Bacteria that enter the cell typically do so through No, I don't want to evolve to kill yet. Still need to finish infecting Greenland. And to do that, going to bump up cold resistance a hair. Yeah, that should help it start spreading through here faster. And down here you can see the progress of cures. It also tells you what percentage of the world has been affected, whether or not you've been noticed, and in this case so far I have not. The status of cure research, what borders have been closed, shipping ports have been closed, airports. So the last 3 countries I have to completely infect are Greenland, Iceland, and Ukraine. Gonna go ahead and bump up cold resistance again. There we go. Now those little red dots are starting to show up. Anyway, the intracellular bacteria, because they reproduce within the host cells, they tended not to produce tissue-damaging toxins. Their intracellular nature also makes it really difficult to eradicate these infections completely, and chronic diseases may result. Early infections by intracellular bacteria can be controlled by the defensins secreted by neutrophils, which can destroy the invaders before they enter the cells. Those that escape the defensins are taken up by neutrophil phagocytosis or activated macrophages. In addition to phagocytosis, macrophages also carry out TLR-mediated endocytosis. There we go, there goes Greenland. So let's go back here, devolve you, devolve you, devolve you, devolve you, devolve you, leave those there for a little bit until this is completely. So infection in Greenland has slowed a little bit because I devolved those, but it is going up gradually. You can see that number down here. Another part of the immune response to intracellular bacteria are the NK cells, which are stimulated by macrophage activity. They detect infected host cells by a deficit in the MHC class expression and destroy them via natural cytotoxicity. In addition, the NK cells produce interferon gamma, a cytotoxin which promotes the activation of macrophages and T helper cell differentiation. Now, CD8 or cytotoxic T cells produce CTLs, which are critical for resolving intracellular infections by relying on secreted TNF and interferon gamma, or granule components with direct antimicrobial activity. There are also CD4 or helper T cells, which contribute to the defense against intracellular bacteria because of the produced interleukin-2. Brexit. Because of produced interleukin-2, which supports T cell differentiation, and Th1 cells are required for macrophage hyperactivation. The interferon gamma produced enhances the macrophage microbial killing abilities. Now, bacterial components released from a dying infective cell also have the potential to activate B cells to produce neutralizing antibodies which bind to newly arrived bacteria or bacterial progeny that have been released. The antibody-bound bacteria are unable to enter host cells and are eliminated by phagocytosis or classical complement-mediated lysis. When an intercellular pathogen avoids death by CTLs and hyperactivated macrophages, the body attempts to isolate the pathogen in a granuloma, which forms around infected macrophages. The inner layer contains CD4 T cells and macrophages, while the outer layer is composed of CD8 T cells. Eventually, the exterior of the granuloma becomes calcified. Let's go here and bump this up again, just to speed up infection here. With the granulomas, no, stop mutating symptoms. Anyway, with the granulomas, like I was saying, the inner layer contains CD4 T cells and macrophages, while the outer layer is composed of CD8 T cells. Eventually, the exterior of the granuloma becomes calcified, and the cells in the middle undergo necrosis. Again, in all cases, these responses are not foolproof. Intracellular bacteria can avoid phagosomal destruction by replicating in non-phagocytic cells, such as the Schwann cells of the peripheral nervous system. Other bacteria intentionally enter the phagocytes and deactivate them by synthesizing molecules which block lysosomal fusion and ROI destruction. They can also recruit proteins created by the host cells to block lysosome function. Why aren't you spreading? Let's go. No, I don't have enough DNA points to bump that up. It's going slowly. Probably shouldn't have reduced them down so quickly for transmission in this area and in Iceland. No, don't want to do that. Might. No, don't really want to take the risk and evolve too soon. Just go ahead and bump up the speed a bit. So, as I was saying, hyperactivated macrophages can be avoided by blocking the expression of the host genes required for hyperactivation, and antibodies can be avoided by spreading to other host cells, limiting the ability of the antibodies to bind. Also, some intracellular bacteria avoid triggering T cell response by interfering with the functionality of antigen presenting cells. Have I infected the entire world? Yes, okay, so the entire population is infected. Now we can move on to the fun stuff. De-evolve you, de-evolve you, come into transmission, start devolving all of this stuff. That will give us some of our points back. So we can move in here and start doing the fun stuff of evolving symptoms. The one that I like to go for is total organ failure. Which we'll see if we can actually hit it before. Okay, so we've caused our first death. No healthy people left in the world. And at this point, it's kind of balancing resources. You want to kill off everybody in the world before the cure effort reaches 100%, because then any progress that you've made has been lost. Which is why I like going for total organ failure, because if you can hit that, it will wipe out a population ridiculously quickly. So for that, we need 31 DNA points. So let's see how this goes. So you can see that they're already starting to shut down airports. This is why you want your disease to remain hidden for as long as possible. Otherwise, people start to shut down modes of transit. And I might lose this one because they're already pretty high on curing. And I've only killed 7 million people. So I might end up losing this scenario. Which does raise an interesting question. If there was ever a global, well, I mean, there have been global pandemics in the past, but with the current state of global politics, would, in the event of a pandemic such as this, would there actually be cooperation with research? Or would it be a case of one country finds the cure and then, it's a case of sell to the highest bidder? Yep, I'm gonna lose this scenario. If I wouldn't have bumped up the cold resistance to hit Greenland and Iceland faster the second time, I probably would have been able to win this, but because I used those 22 DNA points to bump up transmission there, it kind of threw off the rest of the game. Hmm. Yep, there it is. The cure is completed and is being deployed globally. So if you look down here, you can see the infection reduces rapidly. Oh, and my bacterial pathogen has been eradicated. Oh well. So yeah, it was a pretty good run. Yeah, almost 3,000 days. So yeah, anyway, yeah, for anybody that's stuck around this long, and I would honestly be surprised if anyone had, because I probably lost everyone at the beginning of this mess. This ramble was a very, 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 very much a simplification of some of the immune responses to bacterial invaders. I'm by no means an expert in the material, so I may have a few things wrong. Some of this I'm working off of memory. And I might continue with this and do some similar things for some of the other pathogens that one can experiment with in this game, but I don't know yet. I'll just have to see if I can get a better working format together, because I don't know, this recording felt a little sloppy, if you will. Anyway, I don't know. If this is something that you would be interested in seeing, then feel free to leave a comment below, or, comment saying that, no, this was awful and I should never do this again, and maybe I'll listen, maybe I won't. Either way, Arashi Kitsune signing off.